Alright, everyone. So we have gone through the basic steps of glycolysis. Now we're going to talk about its regulation. Whenever we talk about regulation, there are basically two types of regulations, and one is allosteric regulation and the second one is hormonal regulation. Well, if we talk about allosteric regulation, let's take an example of an enzyme. Alright, this is an enzyme. Just take an example of that. And here's the active side of this enzyme. Now the substrate is going to attach on this active side and produce a reaction. However, there is another side known as allosteric side. Now this allosteric side, in here there are certain compounds that are going to attach and they're going to influence the enzyme activity. Either they will be increasing the amount of reaction or either they will be decreasing the amount of reaction acting on the enzyme, certain enzyme. Alright, so there is a regulation, allosteric regulation, and there is then hormonal regulation. So if we specifically talk about the hormonal regulation of glycolysis, then it is mostly regulated by insulin and glucagon. And insulin is always there when there is a high amount of glucose in the blood, and whenever glucagon is there when there is low amount of glucose in the blood. And you can also remember it by glucagon, glucose, gone, glucagon. Alright, so when there will be less glucose, there will be action of glucagon, and when there will be more glucose, there will be action of insulin over here. So these both regulations are going to occur in this glycolysis pathway, which we're going to discuss over here. So here is the 10-step reaction of glycolysis. You can see it clearly over there. And there are three main enzymes that are highly regulated in this process, which are known as hexokinase, phosphofructokinase, and pyruvate kinase. These three enzymes are highly regulated over here, and we're going to discuss them one by one. So firstly, we're going to talk about hexokinase. Hexokinase is also known as glucokinase over here. Hexokinase is also known as glucokinase when it's inside the liver. So it stays inside the liver in the form of glucokinase, and other than that, in all other parts of the body, it's known as hexokinase, and it is present in the form of hexokinase. Alright. So what happens here is that whenever there is need of glycolysis to occur, when the cells want more glycolysis, when they want more energy, then what they're going to do? They're simply going to regulate this glucose. They're going to ask glucose to increase the amount of hexokinase. And this will be a plus reaction. This will be increase of hexokinase inside the body. And what if we don't want glycolysis that much? What if there's too much increase in the ATP? Then what will happen? That glucose 6-phosphate is going to downregulate this enzyme, and the hexokinase activity will be lowered. So what happened here is that this is the allosteric regulation I'm talking about. So what about the glucokinase that is present in the liver? So glucose is going to act over here. Glucose is going to drag that glucokinase out of this liver, and this is going to increase the amount of glucokinase and hexokinase, which will increase the amount of, obviously, glycolysis. And what if there's too much glycolysis and we want to reverse all of this? So for this purpose, fructose 6-phosphate is going to act, and it will just reverse its role, and glucokinase amount will be reduced due to fructose 6-phosphate. Alright, so this is pretty simple about the allosteric regulation. So if we talk about the hormonal regulation, there will be involvement of insulin and glucagon. And if we talk about insulin, there will be insulin whenever there's an increased amount of glucose. Alright, so whenever there's increased amount of glucose, the hexokinase and glucokinase are given signals to make more of these enzymes so that there is more glycolysis going on. Obviously, there is more glucose, so glycolysis should also be like it increased in number. And what if there's low level of glucose? What if there is no glucose inside the blood right now? So glucagon is going to activate itself, and it's going to tell hexokinase and glucokinase that you need to stop, you don't need to multiply that much because we don't need more of the glycolysis because there's already low glucose level in the blood. Alright, so this glucagon is going to negate the processes of hexokinase and glucokinase. Alright, so this was all about the regulation of hexokinase enzyme. So let's move towards the regulation of phosphofructokinase enzyme. This is the third step of our glycolysis, and that's how it's going to act. So what happens here is that whenever there is increase in ATP or there is much more glycolysis going on over here. So the ATP is going to signal phosphofructokinase to slow down the enzyme and slow down the process of glycolysis over here. So all right, so it will be helpful in the down regulation. And citrate is also going to act. Citrate actually is the part of Krebs cycle, and Krebs cycle is also helpful in the generation of ATP. So whenever there is more citrate inside our body or in Krebs cycle, then it will also signal phosphofructokinase to down regulate its activity, so that the process of glycolysis then just can stop. All right, so what about ADP? What will happen when ADP is going to act on phosphofructokinase? Obviously it's going to tell to increase its activity, because whenever there is more ADP, which means there is low ATP. If there is more ADP, that means more ATP is not producing. So that's why it's going to signal phosphofructokinase to increase in number and increase more and make more ATP inside the body. All right. One more tricky thing that happens over here, that in this step where fructose 6-phosphate is converted into fructose 1,6-bisphosphate, most of the reaction is going on over here in the glycolysis. But some of the fructose, very small amount of the fructose, undergo one more reaction with the two enzymes, the combination of two enzymes, and these enzymes are phosphofructokinase 2 and the other enzyme is fructose 2,6-bisphosphatase. Both of these enzymes work as a complex, and they're going to convert fructose 6-phosphate, very little amount of this fructose 6-phosphate, into fructose 2,6-bisphosphate. And this will be occurring due to this first part of the enzyme, which is known as phosphofructokinase 2. All right, so first part of the enzyme is going to convert this fructose into fructose 2,6-bisphosphate. And if we want to convert this fructose 2,6-bisphosphate again back into fructose 6-phosphate, then the other part of the enzyme is going to act, which is fructose 2,6-bisphosphatase. All right, so you've got the point. The very small amount of fructose is converted into fructose 2,6-bisphosphate, and it is then again converted into fructose 6-phosphate, and by the action of this combination of these two complex enzymes. Now what happens here is that this fructose 2,6-bisphosphate is going to upregulate this phosphofructokinase to make more ATP, to make more glycolysis. So it's going to encourage the glycolysis process over here. This it's high regulator of this glycolysis pathway. It is known as this fructose 2,6-bisphosphate is known as the high regulator of glycolysis. All right, so this is the whole process where there are two enzymes, ATP and citrate, are going to act on this phosphofructokinase, and they will say to downregulate the process, and ADP and phosphate 2,6-bisphosphate are going to upregulate the process of making more phosphofructokinase. All right, so this is the allosteric regulation of phosphofructokinase. So what about the hormonal regulation? Okay, it's going to be a little bit tricky, but bear with me for that. So first there is insulin, and then there is glucagon. Whenever there is low levels of glucose inside the blood, so what will happen here, that glucagon is going to upregulate fructose 2,6-bisphosphatase. It's going to upregulate this enzyme. Whenever it's going to upregulate this enzyme, it will in turn upregulate the phosphofructokinase. All right, so and it's also going to downregulate this PFK enzyme. All right, so that there will be no conversion of this fructose into fructose 2,6-bisphosphate. Instead, it's going to convert this fructose 6-phosphate into the main process of glycolysis, which is known as fructose 1,6-bisphosphate. All right, so if we talk about insulin, the insulin is going to upregulate PFK, so there will be more production of fructose 2,6-bisphosphate. All right, and whenever it's going to act on fructose 2,6-bisphosphatase enzyme, it's going to downregulate it, so that there will be less production of this enzyme and conversion of fructose 6-phosphate. All right, so that was a bit tricky to learn, but I hope I've made your concept clear over here. So that was all about the regulation of phosphofructokinase. All right, so moving towards the regulation of last enzyme, which is known as pyruvate kinase, and it is pretty simple to understand. Well, what happens here is that whenever there is increase in ATP, this will going to be stimulate this pyruvate kinase that is going to stop the reaction of glycolysis. And whenever there is long chain fatty acid coenzyme A is involved, they're involved in the production of beta oxidation. They're involved in beta oxidation over here. So what happens here is that they're also going to signal that pyruvate kinase needs to stop making more of the process of glycolysis. So for acetyl coenzyme A, it's also going to downregulate this enzyme because it's also taking part in the Krebs cycle, and Krebs cycle is also helpful in the generation of ATP. All right. So if we talk about, this was the allosteric regulation, and if we talk about the hormonal regulation, glucagon is also going to downregulate this pyruvate kinase because there will be low levels of glucose in the blood. So glucagon is going to signal pyruvate kinase that it needs to stop making glycolysis so that the glucose stays inside the blood. All right. So, but if we talk about insulin, insulin is going to be acting on the increased glucose levels. So it will stimulate pyruvate kinase to make more glycolysis, more of the process of glycolysis. And one more unique compound that is going to act on pyruvate kinase is fructose 1,6-bisphosphate. Now this was the third step of glycolysis, and why it's acting over here. So it's an example of a feed forward reaction, which means that it's so far away from happening, but still it's going to signal this enzyme that we're going to come, they're going to make more of the ATP, we're going to make more of the glycolysis of the process. So it needs to stimulate itself, it needs to stimulate pyruvate kinase so that it can make more ATP and more pyruvate kinase. All right. So this is the example of a feed forward reaction. So that's pretty much it about the regulation of these enzymes, these three important enzymes that were highly regulated inside the glycolysis. So we're done with the regulation. Now we're going to discuss about the conversion of pyruvate to acetyl coenzyme A, which is a very small step, and the regulation for it in the next section. So stick around with us.